The homing endonucleases are a collection of endonucleases encoded either as freestanding genes within introns, as fusions with host proteins, or as self-splicing inteins. They catalyze the hydrolysis of genomic DNA within the cells that synthesize them, but do so at very few, or even singular, locations. Repair of the hydrolyzed DNA by the host cell frequently results in the gene encoding the homing endonuclease having been copied into the cleavage site. Hence the term homing to describe the movement of these genes. Homing endonucleases can thereby transmit their genes horizontally within a host population, increasing their allele frequency at greater than Mendelian rates. Origin and mechanism. Although the origin and function of homing endonucleases is still being researched, the most established hypothesis considers them as selfish genetic elements, similar to transposons, because they facilitate the perpetuation of the genetic elements that encode them independent of providing a functional attribute to the host organism. Homing endonuclease recognition sequences are long enough to occur randomly only with a very low probability, approximately once every 7 times 109 BP, and are normally found in one or very few instances per genome. Generally, owing to the homing mechanism, the gene encoding the endonuclease, the HEG, homing endonuclease gene, is located within the recognition sequence which the enzyme cuts, thus interrupting the homing endonuclease recognition sequence and limiting DNA cutting only to sites that do not yet carry the HEG. Prior to transmission, one allele carries the gene HEG+, while the other does not, HEG-, and is therefore susceptible to being cut by the enzyme. Once the enzyme is synthesized, it breaks the chromosome in the HEG- allele, initiating a response from the cellular DNA repair system. The damage is repaired using recombination, taking the pattern of the opposite, undamaged DNA allele, HEG+, that contains the gene for the endonuclease. Thus, the gene is copied to the allele that initially did not have it and it is propagated through successive generations. This process is called homing. Nomenclature. Homing endonucleases are always indicated with a prefix that identifies their genomic origin, followed by a hyphen, I, for homing endonucleases encoded within an intron, pi, for protein insert, for those encoded within an intein. Some authors have proposed using the prefix F, freestanding, for viral enzymes and other natural enzymes not encoded by introns nor inteins, and H, hybrid, for enzymes synthesized in a laboratory. Next, a capital letter is derived from the first letter of the name of the genus of the natural source organism, and 2 lowercase letters are derived from the name of the species of that organism. Finally, a Roman numeral distinguishes different enzymes found in the same organism. For example, we can mention the enzyme PyTLIII that is the second enzyme encoded by an intein found in the Archaeothermococcus literalis, and H-DRE, the first synthetic homing endonuclease, created in a laboratory from the enzymes IDMOI and ICRE, taken respectively from D-sulfurcoccus mobilis and Chlamydomonas reinhardtii. Comparison to restriction enzymes. Homing endonucleases differ from type 2 restriction enzymes in the several respects. Whereas type 2 restriction enzymes bind short, usually symmetric, recognition sequences of 4 to 8 BP. Homing endonucleases bind very long and in many cases asymmetric recognition sequences spanning 12 to 40 BP. Homing endonucleases are generally more tolerant of substitutions in the recognition sequence. Minor variations in the recognition sequence usually decrease the activity of homing endonucleases, but often do not completely abolish it, as often occurs with restriction enzymes. Homing endonucleases share structural motifs that suggest there are 4 families, whereas it has not been possible to determine simply recognizable and distinguishable families of type 2 restriction enzymes. Homing endonucleases act as monomers or homodimers, and often require associated proteins to regulate their activity or form ribonucleoprotein complexes, wherein RNA is an integral component of the catalytic apparatus. Type 2 restriction enzymes can also function alone, as monomers or homodimers, or with additional protein subunits. But the accessory subunits differ from those of the homing endonucleases. Thus, they can require restriction, modification, and specificity subunits for their action. Finally, homing endonucleases have a broader phylogenetic distribution, occurring in all 3 biological domains: the archaea, bacteria and eukarya. Type 2 restriction enzymes occur only in archaea, bacteria and certain viruses. Homing endonucleases are also expressed in all 3 compartments of the eukaryotic cell: nuclei, mitochondria and chloroplasts. Open reading frames encoding homing endonucleases have been found in introns, inteins, and in freestanding form between genes, whereas genes encoding type 2 restriction enzyme genes have been found only in freestanding form, almost always in close association with genes encoding cognate DNA-modifying enzymes. Thus, while the type 2 restriction enzymes and homing endonucleases share the function of cleaving double-stranded DNA, they appear to have evolved independently. Structural families. Currently there are 6 known structural families. Their conserved structural motifs are LAGLIDADG. Every polypeptide has 1 or 2 LAGLIDADG motifs. The sequence LAGLIDADG is a conserved sequence of amino acids where each letter is a code that identifies a specific residue. This sequence is directly involved in the DNA cutting process. Those enzymes that have only one motif work as homodimers, creating a saddle that interacts with the major groove of each DNA half site. The LAGLIDADG motifs contribute amino acid residues to both the protein-protein interface between protein domains or subunits, and to the enzyme's active sites. Enzymes that possess 2 motifs in a single protein chain act as monomers, creating the saddle in a similar way. The first structures to be determined of homing endonucleases, of PISCEI and ICRA, both reported in 1997, were both from the LAGLIDADG structural family. The following year, the first structure of a homing endonuclease, ICRA, bound to its DNA target site was also reported. GIY-YIG. These have only one GIY-YIG motif, in the N-terminal region, that interacts with the DNA in the cutting site. The prototypic enzyme of this family is ITEVI, which acts as a monomer. Separate structural studies have been reported of the DNA binding and catalytic domains of ITEVI. The former bound to its DNA target and the latter in the absence of DNA. His C-wise box. These enzymes possess a region of 30 amino acids that includes 5 conserved residues, 2 histidines and 3 cysteines. They coordinate the metal cation needed for catalysis. IPPOI is the best characterized enzyme of this family and acts as a homodimer. Its structure was reported in 1998. HNH. These have a consensus sequence of approximately 30 amino acids. It includes 2 pairs of conserved histidines and 1 asparagine that create a zinc finger domain. IHMUI is the best characterized enzyme of this family and acts as a monomer. Its structure was reported in 2004. PD, D, E, XK. These enzymes contain a canonical nuclease catalytic domain typically found in type II restriction. Endonucleases. The best characterized enzyme in this family, ISSP6803I, acts as a tetramer. Its structure was reported in 2007. VSR-like. These enzymes were discovered in the Global Ocean Sampling Metagenomic Database and first described in 2009. The term VSR-like refers to the presence of a C-terminal nuclease domain that displays recognizable homology to bacterial very short patch repair. VSR. Endonucleases. Domain architecture. The crystal structure of the homing endonuclease PISCE revealed 2 domains. An endonucleolytic center resembling the C-terminal domain of Drosophila melanogaster hedgehog protein, and a second domain, homing endonuclease-associated hinterdomain, containing the protein splicing active site. See also REBASE, a comprehensive restriction enzyme database from New England Biolabs with links to related literature. List of homing endonuclease cutting sites. ICRE homing endonuclease. Meganucleases. Restriction enzyme. Introns and inteins. Intragenomic conflict. Homing endonuclease genes. Transposon. Perler FB. InBase. Retrieved 9 August 2010. The Intein Database and Registry. From New England Biolabs. Perler FB. January 2002. InBase. The Intein Database. Nucleic acids Res. 30. 1. 383-4. PMC 99080. PMID 11752343. DOI 10.N93, NAR 30.1.383. This article incorporates text from the public domain PFAM and Interpro IPRO 07868. This article incorporates text from the public domain PFAM and Interpro IPRO 07869.